I thought he said he was.
I looked all the way down that window. This is number 30 of the graduate students who I've mentored to completion. Alyssa, Stasse, well, almost completion, another couple hours. And Alyssa was in the pool of applicants to DBS. And I looked at her application and thought, dang, this person has everything I need for somebody to just jump in and start doing stuff in our lab. And I invited her and she came up and visited. And I'm so happy that she came on. You came from Clemson, right?
Yeah.
And your parents, I think, came with you from New Jersey. And they are in fact here today in the back. So thank you for being with us. So Alyssa and Matt Chang pretty much ran the lab for like a year. And it has worked out great. I'm just really proud. She's going to tell you about several aspects of oysters in Great Bay. And I hope you enjoy what she has to say.
Thank you. All right, so my project here today is called Infection, Etiology, and Larval Assessment of Cultivated and Wild Oysters, which are Crassostrea virginica of the Great Bay Estuary in New Hampshire. I just want to thank everyone for coming today, especially my committee, which is Bonnie, Liz Harvey, and Ray Grizzle. So a little overview of what I'm going to do today. First, I'm going to talk about the background knowledge and give a brief introduction of my topic. Then I'm going to take you through the main objectives, the problems that I was trying to solve. Chapter 1 focuses on the reproductive output, so the larval assessment of Great Bay eastern oysters and how temporal dynamics, so physicochemical factors, plays into that. Chapter 2 then looks at the infection etiology of cultivated and wild oysters. So I'll be looking at 2 diseases, Haplosporidium nelsoni and Perkinsus marinus in Great Bay. I'll be looking at them through the water column in chapter 2A, and then in chapter 2B, I'm going to be looking at how the diseases are in farmed and wild oysters. Then I'm going to be talking about the significance of this study and then follow it up by acknowledgements and questions. So a little introduction. In the 17th century, English settlers came over to the United States and they were greeted with oyster reefs everywhere along the coast. They didn't really realize what the how important these were to the ecosystem yet, so they kind of kept them a local stock, and all the locals in the area would eat the oysters. However, towards the early 18th century, they started to realize the economical importance of oysters, and they began shipping them out from the Delaware and Chesapeake Bays all over the country. In 1886, the first recorded case of oyster farming occurred, and that is where oyster seed, which is the larvae and spat, were purposefully planted for growth. So this is where the farmers started to actually try to produce oysters. However, this kept happening, and because of this and unregulated harvesting, the population started to decline. There was no regulation set, and so they just kept taking oysters and kept shipping them off. Other things that contributed to that were adverse weather, channel dredging, and disease. So once the oyster farmers began to realize the potential, they started shipping them out. In 1919, it was one of the most valuable industries with an annual return to fishers of $15 million. Then it became just a steady industry, was growing still. And in 2015, it was estimated in the USA, the oyster production is worth $234 million. Today, it continues to be really important. Oysters are everywhere and people ship them out from all over the coast. Not only are they important as a financial gain, so like for food and stuff like that, they are also important in terms of ecosystem services. So oysters are bivalve filter feeders. They take their plankton by filtering it through, And then while they're doing that, they're also decreasing the levels of nitrogen pollution in the water. It's estimated that oysters in Chesapeake Bay can filter the entire bay within 1 day. So there's a lot of filtration going on every single day. Not only are they water filters, they are also a habitat for juvenile fish, such as Atlantic cod, bluefish, and white flounder. It protects the younger fish from larger predators, so they're able to hide in those reefs and make sure that they don't get attacked by bigger fish. They also are really important in shore protection. So a study by Brandon et al. In 2016 found a 75% reduction in wave energy with oyster reefs when waves from Hurricane Sandy hit the New Jersey coast. So it's really important in keeping that shore the way that it is and keeping the sand and all the ecosystems that go on the shore intact. So the Great Bay Estuary is where I am doing my research. I am doing this one because it's local, and Great Bay Estuary is a super unique estuary in that the tidal range is 2 to 4 meters, and it can, during the tide changes, change the water up to 50% of the water goes in and out. So what I have here is a map. All of the dark gray shaded regions are natural oyster reefs. The sites that I selected are also shown with diamonds, which are the oyster farm that I used, which is OF in the top corner in Little Bay. AP is Adams Point, and then we have Nanny Island and Woodman Point. Eastern oysters in Great Bay have always been a huge economy, but recently they are about 10% of what they were in the late 20th century. This could be due to a number of things such as overharvesting, stuff that I talked about before, but another thing you can think of is a limited or a lack of substrate for them to set on. So they like to set on stuff such as old oysters, or in our case, we use ceramic tiles. If there is not enough of that material, then they can't set and they can't grow to their full potential. Temporal dynamics have also played a large role in this. So climate change, the weather getting more severe, changes in salinity, pH, and temperature, those are all impacting oysters in Great Bay. So another thing that we are looking at in this study is diseases in oysters. And the first disease that I am looking at is MSX. It was first seen in oysters in Delaware Bay in 1957. And they found that the causative agent of this was Haplosporidium nelsoni. It is a protozoan parasite. It infects the gill epithelium, and the gill epithelium is super important with filtration. So if you don't have that intact gill epithelium, the filtration cannot happen as well as it needs to. It forms systemic infections. So when people are using histology to look at the infections, they actually take a syringe or needle, they stick it in the oyster shell, feel around for the muscle, and then extract the hemolymph from the muscle. And you're able to look at the hemolymph because it is a systemic infection. So that's what's shown here. This is Haplosporidium nelsoni in oysters. So the S is pointing to a spore, and the P is pointing to a plasmodial stage. And so those are the two stages. Right now, they don't know the method of transmission. Ford in 2018 tested tens of thousands of different intermediate hosts and couldn't find any of that. They think the spore stage has something to do with it, but there was not enough information to definitively tell if that is the reason that oysters are getting this disease. The other disease that I am looking at is dermo. It was introduced to the Delaware Bay in the 1950s. However, the water was too cool for it to infect oysters. In the 1990s, after a few winters that were surprisingly warmer than usual, it started infecting the oysters. And they found out that this infection wasn't caused by Haplosporidium nelsoni, it was caused by Perkinsus marinus. So contrary to MSX, dermo's method of transmission is very well known. Dead infected oysters release the zoospores into the water column, and then those are able to infect other oysters. This is mostly concentrated in the rectum of the oysters. So when you are doing the histology for this, you would extract the rectum, put it in a medium to grow and incubate, and then you would look at the cells under a microscope and count the number of cells. So that's what you can see over there. There are a bunch of those purple cells, and those are the P. marinus cells in an oyster. I'm now going to talk about my project goals. So the first one that I wanted to look at the reproductive output of eastern oysters in Great Bay by assessing the larval abundance and determining if physicochemical factors are associated with the abundance in Great Bay. Then I wanted to focus more on the disease because there's a million different things that we can contribute to oyster population decline, but I wanted to just look at the disease. And so I wanted to quantify the infection of H. nelsoni and P. marinus in oysters and also in the water column using PCR-based methods. And then I wanted to look at the infectious rates of H. nelsoni and P. marinus in farmed and wild oysters to see if there was a difference between the two types. The first thing I'm going to be talking about is the reproductive output, so the larval assessment compared with temporal dynamics, so the physicochemical factors of eastern oyster larvae. And just reiterating a little bit, I want to see if there is anything that is going on with the larvae, if you can narrow it down to like one thing that's going on based on the physicochemical factors. I also want to see if there's any differences across the bay as a whole. So if there's a differences between wild and farm reefs. And I want to do this because no studies so far have provided this baseline understanding of anything. So what I did is I went out in the field, as you can see here, and I put out spat collectors. They were a cage and they had 3 ceramic tiles in them. We put out 3 per site and there were 4 sites. All of the tiles were approximately 15 meters apart. And while we were doing that, we also did larval tows every week. So we took a bongo net, we put it under the water approximately 0.3 meters, carried it around for 2 minutes, going approximately 2 knots. And then we got a sample of water that had the plankton species and the oyster larvae that we were hoping to see in there. We then went into the lab, filtered out the water through a set of brass sieves, and they were selected for 45 to 106 microns, which is where oyster larvae fall. We all did 3 1 mil replicates and looked at those under a microscope with 4x magnification. You can see in the top right picture that there is a veliger, which is the second stage of oyster development. And we counted the number of veligers and dehinge per 1 mil replicate. To get a more accurate understanding, because microscopes are not always the most accurate, we also did this within the flow cam. So we did 3 5 mil replicates through the flow cam with a 600x flow cell. And we were able to create a known library of oyster larvae. And once we ran our sample through, the library sorted them out based on how similar they were to the known oyster larvae. And then we were able to select and go through every single one to see if that was an oyster larvae. So that is what you see in the top left picture. That is a dehinge, which is the first stage of oyster development. And so once all this was done, we had a rough estimate count and we all went through, me and 2 other people, to make sure that what we were seeing was in fact oyster larvae. And we had to have 2 out of the 3 people agree that it was in order for it to count as an oyster larvae. We then got physicochemical data from the Nears buoy in Great Bay estuary, and we collected pH, salinity, and temperature. Studies have shown that this is representative of Great Bay as a whole, so we were able to use that as an overall indicator. And this is just the physicochemical data plotted out. So you can see temperature, as expected, was high in July, went up a little bit in August, and then went down. Salinity varied a little bit, and pH was kind of around the same throughout the whole entire time. So when I was looking at the data, I realized that my data was not normally distributed, and I wanted to try to get it normally distributed. So what I did was use a log x plus 0.05 transformation, and I found that that did meet the assumptions for normality and homogeneity. So then I ran ANOVAs between the sites to see if there was any differences between the sites. After that was done, I ran a Tukey's test to see differences between the sites and the dates, see if there was any differences across the dates. The spat that I found on the ceramic tiles, I was not able to get that data normally distributed. So I used a Kruskal-Wallis test to test for differences in the number of spat among dates. And then I used regression models for the physicochemical data. And this is what I found. So this is just all the data plotted out. On the left, we have the D-hinge larval counts, and on the right, we have veliger larval counts. I found that there was no differences between the sites. All of the sites were essentially the same and not significantly different. So I was able to use those counts as a whole for Great Bay, showing that there was no differences among the Great Bay as a whole. When I ran the regression models on physicochemical factors, I found that temperature was the only physicochemical factor that was significant. It was significant in both D-hinge and veliger larval counts. So that is that plotted out. As temperature increases, the number of veliger and D-hinge larvae also increase. And then here is the spat settlement data. I found that there were significant differences in spat settlement. And we found that July had the highest amount of spat settled, but we were seeing them as early as June, suggesting that there was a spawning activity occurring previously than we thought, which was, we thought it was happening in May or June. It's probably happening in April or May. Settlement, though, was not great, and that is because a lot of tunicates were on all of the tiles. So as you can see on the left, the tile is completely covered in tunicates, and there was no areas for the spat to settle. So some key takeaways from this chapter is, first and foremost, there are not enough larvae to sustain the wild populations. There is not enough healthy oysters producing the larvae that we need to keep oyster populations where they need to be. Restoration efforts in Great Bay should consider other factors than simple physicochemical variation. They should look at the phytoplankton abundance and composition. They should look at predation and substrates that are in Great Bay for oysters. It is also important to begin sampling by April or possibly earlier, as the average turnaround from spawning to settlement is 32 days. So we want to, since we saw settlement in June, we want to get there before that is happening. Some future studies that would benefit from this are hydrodynamic modeling to see how the oyster larvae are moving in Great Bay and see how they're settling. Even though we know there's no differences with sites, I want to see how they're moving throughout the bay. So now moving on from the reproductive aspect of oysters, I wanted to look at infections in oysters. And so I looked at dermo and MSX, specifically H. nelsoni and P. marinus, which are the causative agents. And first, I wanted to see what they were like in the water column, as if they're more concentrated in certain places than others, you would try to avoid those places for oyster restoration and putting oysters out there. Then I wanted to use histological samples that I had from the Rutgers Haskin Shellfish Lab from Emily McGurk and Dave Bushek. They had their oysters classified by histology, and I wanted to see if I could use QC-PCR, which is quantitative competitive PCR, to match those histology classifications. Finally, I wanted to compare the two groups of oysters, so wild and farmed, and see if there was differences in the amount of infectious agent between the two. So the first part of this is where I am assessing H. nelsoni and P. marinus in the water column as a whole. I took those water samples that I had from the larval toes and I filtered them through a 2-micron filter using vacuum filtration. And I took that filter and rolled it up into like a tiny little ball and then shoved it in a tube and did the DNA extraction using a DNA Easy Power Water Kit. Once I had that completely done, I had approximately 100 mils of extracted DNA, and the average concentration for all the samples was 7 nanograms per microliter. I quantified this using the tape station. As you can see, this is an example of using the tape station. So it tests for quantity and quality of the DNA. I have a thick band up there showing that the DNA is not fragmented. It's in one solid piece, which is really important for later use. And I was able to know the exact concentration. So once I did that, I could standardize the concentration to 5 nanograms per microliter. This is important in PCR because you need to make sure that all the concentrations are the same for future analysis. And so I did 5 nanograms per microliter because my DNA the PCR protocol I used called for 25 nanograms per microliter, so I was able to use 5 mils instead of 1 mil since the DNA concentration was so low in water samples. These are the primers I used. I used CVI-11, which is a microsatellite in oysters, and that was to detect oyster presence. That was simply just, I wanted to make sure that they were actually there, kind of a, to make sure that what we were seeing in the water samples through the microscope was actually oysters. So that was kind of like to just double check ourselves. And then I used HNPMA and HNE-PSP, which are 2 primers that can allow dual detection and eventually quantification of H. nelsoni and P. marinus. I then, since I was using QC-PCR, had to generate a competitive sequence. And luckily, I had Will Brooks, who is a former Brown graduate student, who started and finished the work of developing that assay, and I just had to kind of pick up where he left off. So he used a competitor that is a similar size to H. nelsoni and P. marinus, which is super important because you want to be able to see them on the same gel in roughly the same area. We used HNPMA and HNE-PSP-BC primers with known infectious DNA that we know had MSX and dermo in it. We ran it in a thermal cycler under a PCR protocol, and we were able to get this competitor sequence, which it was 281 base pairs. We then quantified that on the tape station, and I was able to dilute the competitor to those serial dilutions, which is important later on when I'm analyzing the data. So once it was run and run through the gel, it kind of looked like this. So this is one with 4 replicates instead of 5. I just used this image because it has a really good banding pattern to show you. So every single sample that I had was run in a replicate of 5 with the 5 serial dilutions. And so you can see in here at the top is where the dermo band shows. The next band down is MSX, and then at the bottom is the competitor, and I labeled the competitors across with the different dilutions. When I was analyzing this, I had to use Adobe Photoshop to find the integrated density, which will show me how bright those are, which correlates to how much DNA is in there. Where the zone of equivalence was intermediate between 2 adjacent lanes, interpolation was employed to determine the competition equivalence point for each parasite. So once I had all this data, I used the concentrations that I got and classified them as either from a farm or from a reef. And then I corrected them for filtration volume and the dilution volume that I used so I could get an accurate representation of how much DNA was there. I did a Kruskal-Wallis test between the 2 groups to determine if there was any differences between each infectious agent at farms and reefs. And then I used a Wilcoxon ring test to determine if there was any differences between the 2 parasites as a whole throughout Great Bay. So if one was more abundant than the other. And this is what I found. So this is all of the 2 data sets, so the H. nelsoni and P. marinus plotted out regardless of reef or natural reef or farm. So I found that while H. nelsoni seemed to have some peaks, which are more abundant than P. marinus, there was no significant differences between the 2 diseases. So in the water column, those diseases are acting the same, they're moving the same, and they're at approximately the same levels. When I looked at the 2 diseases at 2 different types of oyster habitats, so farms and reefs, I found that there was no significant differences between farms and reefs for H. nelsoni and P. marinus. So even though there are varying levels of diseases in oysters at farms and reefs, there are not varying levels of those infectious agents at the farms and reefs in the water column. So now I'm going to dive into looking at H. nelsoni and P. marinus in wild and aquacultured eastern oysters of Great Bay. So like I said, I had oysters that I got from Ray Grizzle and Kristen Ward, and they provided me with a sample of about 10 wild and aquacultured oysters. I just wanted to do a small pilot study for this just because I wanted to see if there was any differences out there. Prior knowledge shows that we know farmed oysters are from an MSX tolerant stock. However, if the oyster larvae is moving throughout Great Bay as a whole, I wanted to see if there was any reproduction, like crossbreeding between the wild and the aquacultured oysters. So I had a small sample from June 2020, and then I got tissue samples from Emily McGurk and Dave Bouchek at Rutgers that gave me MSX and dermo loaded samples, and they classified them out for me so I could run QC-PCR and compare those results to the histology results. I used the DNA extraction, quantification, PCR, and QC-PCR, all the same way that I did in the previous study. So for chapter 2A, I used the power water kit to extract the DNA. I used the tape station to quantify the DNA, and I used the same primers to run the PCRs and the QC-PCRs. The only difference is that these samples had more DNA than the water samples did, which makes sense because it was coming straight from the oyster compared to a whole water sample. And so the average concentration of these were 100 nanograms per mic. I diluted this down to 25 nanograms per mic because that is all I needed in my assay. I then classified these results as no infectious agent, which I set those limits on low, medium, and high, so that I could compare those limits to the histology classifications. I ran a Kruskal-Wallis test between the wild and aquacultured oysters for each infectious agent to determine if there was any differences between wild and aquacultured. And this is what we were looking at when I just laid out all the data on a table. So I found that in the 6 samples that I got from the Rutgers lab, MSX was classified as the same every single time. So where the histology was low, the QC-PCR classification would also be low. However, when I was testing for P. marinus, I blended the whole oyster up when histology only looks at the rectum. And I did the whole oyster because it's a little bit easier to do and you get more of a sample in there. And so I wanted to, when I was looking at this, I realized that my QC-PCR classification was almost always lower than the histology classification. So there is some factor that needs to be added to the QC-PCR to get it to match the histology classification. I have a rough idea of what it is right now just from these 6 samples, but more samples will be needed in the future to get this down to the precise measurement, and we'll be able to implement that and get the same classifications. This is the data when I plotted it out. So in A, I have all the data shown. And since there were a few really high numbers, I also plotted them without those numbers to get a better look at the tiny ones. And so I found that it was mostly accurate, and knowing what I know about dermo and how it's infected in the rectum mostly, I can say that with this weighted factor, it should be accurate 100% of the time in the future. Then when I looked at infectious agents in wild and farmed oysters, I found that the MSX tolerant stocks that are at the farms are MSX tolerant. So while there is a low level of MSX in them, it's not enough to form a systemic infection. And so they're still able to be used in sale and stuff like that. For dermo, I found that both oyster populations had really high levels of dermo. So what we are seeing here is that MSX is under control, but we can't figure out dermo yet. Key takeaways from here is that while there are different levels of the infectious agents in the 2 types of oysters, there is the same levels throughout the water column. So location should not matter here, whether you are planting at one end of the bay or the other. This work also demonstrated the ability to accurately diagnose H. nelsoni, and future work needs to be done to determine the adjustment weight for P. marinus detection with QC-PCR. MSX tolerant strains there are MSX tolerant, which is super important to know. And the next challenge for aquaculturists and biologists in this area is to get a dermo-tolerant strain. Now moving on to the significance and future implications. So for chapter 1, the reproductive output and larval assessment, data suggests rapid climate change can be driving the low numbers of oyster larvae. Since waters are getting warmer, the oyster larvae should be reproducing more, but they can only reproduce up to a certain temperature. If it gets too hot, it's like a hot sauna. You're not going to have the reproduction that you need. So there's that magical window of the best temperature and conditions that these oysters need in order to survive and reproduce. The current data that we have are an important baseline for the eastern oyster larval dynamics. So it will show us how everything is changing, how the temperature, pH, and salinity are affecting this. And future studies will help to determine whether the larval trends are just for this area or can be spread out throughout the entirety of the East Coast where eastern oysters are. For chapter 2, even though there are varying levels of diseases in the eastern oysters, like I said before, similar amounts of the infectious agents are found throughout the water, suggesting that it's not site-based, it is the types of oysters that are at the site. Knowing that MSX is low in aquaculture populations is super important because then we can focus on dermo and getting that levels down. Future work should work on a cross between MSX tolerant and dermo tolerant oysters to increase the genetic diversity. It will slightly lower the resistance to both diseases, but the genetic diversity will increase, causing a bigger population. And if something happens, like a rare outbreak of whatever, just like COVID happened to us, they will have the genetic diversity to deal with it. So that is the data that I had. I would like to thank all of the people here. So Bonnie Brown, Liz Harvey, and Ray Grizzle for everything since I've been here, for field and site help, Kelsey, Grant, Kristen Ward, Laura Brown, Matt Chang, Taja, Alex, my dad, Chloe and Alia. I would like to thank help with funding from Hatch and the Marine Bio Graduate Student Grant. For data analysis and figure help, Patty Jurema, Richard Smith, Chris Neefus, Matt Chang, Carl, and my friend Andy Ackerman. Lab work help, Lauren, Talianne, Shai Meh, Bo Young, Kelsey, Grant, Matt, Taja. And then just for logistics and writing help, Jess Volker and Lisa. And just thank you everyone for helping me throughout this entire process. Does anyone have any questions?
Not even. I think there's some maybe questions in the chat. I don't.
Let's see. Okay, no questions in the chat yet, but question. Easton White said, how do you know the number of larvae may not sustain wild populations? So I just recently published a paper of a multi-year study from 2018 to 2020, and I also have the 2021 data that is not yet incorporated. And the trends of oyster larvae are dramatically going down. So we know that there might be a threshold that the oyster larvae need to sustain this population, and it's just not there because we are not seeing as many matured oysters as we've once seen.
Can you add to that, please, what you learned about looking at studies in other estuaries and their numbers of larvae versus?
Yeah, so other estuaries, I looked at Chesapeake Bay, Delaware Bay. I also looked at stuff in the Gulf of Mexico, and their number of larvae were, as they were adding more larvae, they obviously got a lot more settlement, and those were able to mature into large oysters. And once there were enough large oysters, the reproductive output was so much more. So they didn't have to keep purposefully planting those oyster seeds. And so once there's enough large oysters, we're going to have those populations sustained where they can keep reproducing. Yeah.
So you mentioned that you were looking at oyster stat from 2018 up to present. Is there any historical data on Great Bay oyster stat numbers?
Spat numbers? I don't believe so.
Or dehinge larval?
Dehinge and those numbers, yes. And they are going down dramatically. So I saw data from, I believe, 2012, and the numbers were tenfold the time what they are now.
Okay.
Yeah.
What are like the general infection rates for those two parasites? And is there like a lot of co-infection also? I noticed in the one plot in the farm, like the MSX was really low, but then the other one was higher. Is that like?
So in wild populations, the co-infection rate is through the roof. So both infections are just wildly abundant. However, in the farmed oysters, MSX, they're MSX tolerant, so you'll see those really low levels if you see them at all. And the dermo in those populations is really high. The average that I saw was there were not like systemic infections yet with MSX. It was about like 0.02 nanograms per microliter of infectious content in the samples that I had, and for both of them. So we're seeing a lot of it, which is high for those numbers.
Has there been any historical dips in wild populations, like maybe before the development of a resistant population?
Yes, there have been. That was mostly due to overharvesting and changing like channel dredging, stuff like that. So at first they didn't have any regulations with anything. So they were just harvesting, not realizing that there had to be a certain number of oysters there to sustain the populations. It took a dip and then regulations got put into place for them to stop harvesting every single oyster they saw, and the numbers were able to go back up. There's been some epizootic outbreaks throughout the past, but none that are significant as MSX dermo, Vibrio is another one, stuff like that, yeah.
Were any of those dips sort of like on the scale of this one? Like were they able to recover from something that appears to be this severe?
Yes, the overharvesting one was.
One more question.
Yeah.
You mentioned the protozoans, that one of them had the life cycle worked out. The other one, not so much, but you have the DNA on it. So could you start searching around in the water column and sediments and things like this to try to fill in that gap?
Yeah, definitely. That's been going on for a really long time. And the thought was that there was an intermediate host, so something else in between that's contributing to the oysters passing it. They know that it's not oyster to oyster. So what we really need to focus on is what is going on in between. Like what's that intermediate step? The Ford study that I talked about in 2018 looked at a bunch of different phytoplankton, different things that they were eating, and they could not find any significant transmission between the two. So I think a really cool study in the future would be maybe looking at the sediment, and seeing if there are significant amounts of disease in the sediment, and maybe that is contributing to the oysters passing this disease along. Kelsey asked me, do I like to eat oysters? No. And then I had another question. Are oysters in decline over microplastics and pollution? Yes. Dr. Brown's lab. She looks also at microplastics. Matt Chang has looked at microplastics a lot. I know there's quite a few people at UNH. That's personally not what I do, but I know that they have found microplastics in oysters and different things. And so, yes, that is definitely a cause.
But no physiological
Yeah.
effects that we're aware of. Is that it?
I think that's it.
Well, so the way this works is, first we thank everybody for, where's the, okay. There's some snacks back there, I think, that we forgot to mention.
Yes.
We thank everybody for joining us for this part where Alyssa shows a public audience what she's done. After you guys get some snacks, you get to leave, and the committee will stay here and grill her and decide whether or not she gets to have the degree. And no, we're not gonna. Well, yes, we're gonna grill her, but we'll give her hints and things that she might need to do on her paper copy. And then we make our decision without her here, and then we call her back. So if anybody wants to sit outside for a couple hours, that's fine. And she'll be out eventually. So thank you for driving all the way from New Jersey, and thank you everybody else for taking time out of your busy schedule and for asking very intelligent questions.
Thank you, everyone.
Thank you.